Good afternoon and welcome to this event on microbial identification. And during this presentation, I'll be focusing on microbial identification typing in biopharma. I would like to take this opportunity to thank each one of you for your attention and attendance here, and the organizers for kindly inviting me to speak at this event. So as the title of my talk indicates, I'll be talking about microbial identification methods in the the larger pharma industry. And it will be an overview of the various methods used and looking a little bit into the future and for microbial identification. So microorganisms in biopharma, meaning microorganisms which can be found in pharmaceutical ingredients, including water for pharmaceutical use, the manufacturing environment, intermediates, and finished products, frequently identified to the either the genus level or the genus and the species level, basically to demonstrate microbial control, to exclude objectionable organisms, as in the case of non-sterile products, or absence of microorganisms, as in the case of sterile products. And in the case of environmental monitoring, it is to maintain a level of microbial control in the environments that are used to manufacture these products. There are various microbial identification technologies or systems available which are based on different technologies, different analytical techniques, and each of these methods has its own advantages and disadvantages. And you would need to make a decision on what kind of identification method or technology is appropriate for your use. In some instances, it may be necessary to identify to the strain level, whereas in certain other cases, a broad level identification to the genus level may be sufficient. So with these variables in mind, you will have to use the appropriate microbial identification system. So looking at that, looking at the various types of microbial identification technologies available, obviously, as I said, you can have technologies that will help you identify to the genus or the strain level. They have complexities and associated details of information that you can gather from these technologies, and obviously they are also related to cost, the most expensive being obviously strain typing, and the other ones are microbial characterization, which are simply looking at morphology and staining procedures, looking at phenotypic characteristics, and also more advanced DNA-based or nucleic acid-based technologies. The need for microbial identification is also well established in regulations. For example, FDA guidances, which clearly says the routine identification of microorganisms to the species level or genus level as appropriate, and also suggests FDA recommends wherever possible that nucleic acid-based methods be used, especially for investigational purposes. And also correspondingly in the Pharmacopeia, for example, the USP has in its test procedures and requirements the need for identifying microorganisms, be it non-sterile products and the topic of objectionable organisms, or sterility tests in the absence of microorganisms, where you could use advanced technologies to identify to the species level, especially when you have to invalidate a sterility test. Let's very briefly look at characteristics of available technologies. Phenotypic methods, as the name indicates, looks at phenotypic characteristics such as culture, morphology, physiological properties, biochemical properties, and ability to grow on specific types of media. and proteotypic characterization, which is based on the analysis of stable cellular protein content and using associated mass spectroscopic methods to identify the specific protein in those fragments. DNA-based methods, as I said, are advanced methods, and you have either genotypic methods or phylogenetic methods, which could use either DNA probes or restriction enzyme fragment patterns. In the phylogenetic methods, for example, utilizing typically the 16S RNA gene sequencing for bacteria and the 18S and 28S RNA gene sequences for fungi. Genotypic methods are considered more reliable and advanced because they're based on nucleic acid sequences which are highly conserved. And as I briefly alluded to, these are more expensive than typical phenotypic methods. And for this reason, genotypic methods are frequently outsourced and conducted in contract laboratories. But more recently, advances in technology, also availability of this technology from based on different modifications of the genotypic methods has enabled the availability of scalable methods of genotypic identification, such as the nanopore technology, Oxford nanopore technology, which uses nanopores, and this technology is available in various scales, and this could be brought in-house for microbial identification in real time. Strain typing is needed when you have to identify, when you have to ensure, or when you have to make sure that the genus and the species of the microorganism that you identified in a particular product is what is associated with a particular event, be it operator related or equipment related, and you have to invalidate the test in question, or also in case of epidemiological studies, disease or disease origin, confirming the source of certain disease organisms, where you need to identify and ensure the genus and species of that particular microorganism. And here again, nanopore technology, which is one of the more advanced methods of DNA sequencing, could enable the insourcing of strain typing within biopharma. Polyphasic approach is utilizing various types of information for microbial identification, especially phenotypic and genotypic methods, and building the concept together, not relying merely on phenotypic data or genotypic data, and looking at information that is gathered from all these various methods of identification to get a robust, reliable microbial ID. Issues with current ID and typing methods. In general, current methods require a number of cells, large number of cells. They are you need to grow them and isolate pure monoclonal cultures, and also importantly, they are not suitable for generating long genomic sequences, reading long genomic sequences. Results, importantly, are not generated in real time, or not fast enough, difficult to scale. They're outsourced typically, and currently restricted. Genotypic methods are restricted to critical investigations, and the information in databases are also lacking. So looking at the future, current practices, as I said, include a combination of phenotypic or proteotypic method in combination with genotypic method. Genotypic methods are not currently used as the first line of action, but as I said, recent advances in technology is making it possible for using genotypic methods more widely, and also bringing them in-house, and also for direct reads of short to ultra-long fragments. And here again, I want to point out the use of nanopore sequencing, Oxford nanopore sequencing, and this is particularly going to be helpful for pathogen identification or investigations in case of sterility test failures. Thank you for your attention, and we will be available for questions at the appropriate time. Thanks.